Biochemical oxygen demand or B.O.D. is the amount of dissolved oxygen needed by aerobic biological organisms in a body of water to break down organic material present in a given water sample at certain temperature over a specific time period. The term also refers to a chemical procedure for determining this amount. This is not a precise quantitative test, although it is widely used as an indication of the organic quality of water. The B.O.D. value is most commonly expressed in milligrams of oxygen consumed per litre of sample during 5 days of incubation at 20 degrees Celsius and is often used as a robust surrogate of the degree of organic pollution of water. B.O.D. can be used as a gauge of the effectiveness of wastewater treatment plants. It is listed as a conventional pollutant in the U.S. Clean Water Act. B.O.D. is similar in function to chemical oxygen demand, in that both measure the amount of organic compounds in water. However, COD is less specific, since it measures everything that can be chemically oxidized, rather than just levels of biologically active organic matter. Background: most natural waters contain small quantities of organic compounds. Aquatic microorganisms have evolved to use some of these compounds as food. Microorganisms living in oxygenated waters use dissolved oxygen to oxidatively degrade the organic compounds, releasing energy which is used for growth and reproduction. Populations of these microorganisms tend to increase in proportion to the amount of food available. This microbial metabolism creates an oxygen demand proportional to the amount of organic compounds useful as food. Under some circumstances, microbial metabolism can consume dissolved oxygen faster than atmospheric oxygen can dissolve into the water or the autotrophic community can produce. Fish and aquatic insects may die when oxygen is depleted by microbial metabolism. Biochemical oxygen demand is the amount of oxygen required for microbial metabolism of organic compounds in water. This demand occurs over some variable period of time depending on temperature, nutrient concentrations, and the enzymes available to indigenous microbial populations. The amount of oxygen required to completely oxidize the organic compounds to carbon dioxide and water through generations of microbial growth, death, decay, and cannibalism is total biochemical oxygen demand. Total B.O.D. is of more significance to food webs than to water quality. Dissolved oxygen depletion is most likely to become evident during the initial aquatic microbial population explosion in response to a large amount of organic material. If the microbial population deoxygenates the water, however, that lack of oxygen imposes a limit on population growth of aerobic aquatic microbial organisms, resulting in a longer-term food surplus and oxygen deficit. A standard temperature at which B.O.D. testing should be carried out was first proposed by the Royal Commission on Sewage Disposal in its 8th report in 1912. An effluent, in order to comply with the general standard, must not contain as discharged more than 3 parts per 100,000 of suspended matter, and with its suspended matters included must not take up at 65°F more than 2.0 parts per 100,000 of dissolved oxygen in 5 days. This general standard should be prescribed either by statute or by order of the central authority, and should be subject to modifications by that authority after an interval of not less than 10 years. This was later standardized at 68°F and then 20°C. This temperature may be significantly different from the temperature of the natural environment of the water being tested. Investigators also decided to eliminate anaerobic conditions. Although the Royal Commission on Sewage Disposal proposed 5 days as an adequate test period for rivers of the United Kingdom of Great Britain and Ireland, longer periods were investigated for North American rivers. Incubation periods of 1, 2, 5, 10 and 20 days were being used into the mid-20th century. Keeping dissolved oxygen available at their chosen temperature, investigators found up to 99% of total B.O.D. was exerted within 20 days. 90% within 10 days, and approximately 68% within 5 days. Variable microbial population shifts to nitrifying bacteria limit test reproducibility for periods greater than 5 days. The 5-day test protocol with acceptably reproducible results emphasizing carbonaceous B.O.D. has been endorsed by the United States Environmental Protection Agency. This 5-day B.O.D. test result may be described as the amount of oxygen required for aquatic microorganisms to stabilize decomposable organic matter under aerobic conditions. Stabilization, in this context, may be perceived in general terms as the conversion of food to living aquatic fauna. Although these fauna will continue to exert biochemical oxygen demand as they die, that tends to occur within a more stable evolved ecosystem including higher trophic levels. The B.O.D. test, there are two commonly recognized methods for the measurement of B.O.D. Dilution method. This standard method is recognized by EPA, which is labeled Method 5210B in the standard methods for the examination of water and wastewater in order to obtain B.O.D. Dissolved oxygen concentrations in a sample must be measured before and after the incubation period, and appropriately adjusted by the sample corresponding dilution factor. This analysis is performed using 300 ml incubation bottles in which buffered dilution water is dosed with seed microorganisms and stored for 5 days in the dark room at 20 degrees Celsius to prevent dew production via photosynthesis. In addition to the various dilutions of B.O.D. samples, this procedure requires dilution water blanks, glucose-glutamic acid controls, and seed controls. The dilution water blank is used to confirm the quality of the dilution water that is used to dilute the other samples. This is necessary because impurities in the dilution water may cause significant alterations in the results. The GGA control is a standardized solution to determine the quality of the seed, where its recommended B.O.D. concentration is 198 mg per litre or plus or minus 30.5 mg per litre. For measurement of carbonaceous B.O.D., a nitrification inhibitor is added after the dilution water has been added to the sample. The inhibitor hinders the oxidation of ammonia nitrogen, which supplies the nitrogenous B.O.D. When performing the B.O.D. test, it is conventional practice to measure only B.O.D. because nitrogenous demand does not reflect the oxygen demand from organic matter. This is because B.O.D. is generated by the breakdown of proteins, whereas B.O.D. is produced by the breakdown of organic molecules. B.O.D. is calculated by where is the dissolved oxygen of the diluted solution after preparation, is the DO of the diluted solution after 5-day incubation, is the decimal dilution factor, is the DO of diluted seed sample after preparation, is the DO of diluted seed sample after 5-day incubation, is the ratio of seed volume in dilution solution to seed volume in B.O.D. test on seed, manometric method. This method is limited to the measurement of the oxygen consumption due only to carbonaceous oxidation. Ammonia oxidation is inhibited. The sample is kept in a sealed container fitted with a pressure sensor. A substance that absorbs carbon dioxide is added in the container above the sample level. The sample is stored in conditions identical to the dilution method. Oxygen is consumed and, as ammonia oxidation is inhibited, carbon dioxide is released. The total amount of gas, and thus the pressure, decreases because carbon dioxide is absorbed. From the drop of pressure, the sensor electronics computes and displays the consumed quantity of oxygen. The main advantages of this method compared to the dilution method are simplicity, no dilution of sample required, no seeding, no blank sample. Direct reading of B.O.D. value. Continuous display of B.O.D. value at the current incubation time. Dissolved oxygen probes, membrane and luminescence. Since the publication of a simple, accurate and direct dissolved oxygen analytical procedure by Winkler, the analysis of dissolved oxygen levels for water has been key to the determination of surface water purity and ecological wellness. The Winkler method is still one of only 2 analytical techniques used to calibrate oxygen electrode meters. The other procedure is based on oxygen solubility at saturation as per Henry's law. Though many researchers have refined the Winkler analysis to dissolved oxygen levels in the low ppb range, the method does not lend itself to automation. The development of an analytical instrument that utilizes the reduction-oxidation chemistry of oxygen in the presence of dissimilar metal electrodes was introduced during the 1950s. This redox electrode utilized an oxygen-permeable membrane to allow the diffusion of the gas into an electrochemical cell and its concentration determined by polarographic or galvanic electrodes. This analytical method is sensitive and accurate to down to levels of a plus or minus 0.1 milligram per liter dissolved oxygen. Calibration of the redox electrode of this membrane electrode still requires the use of the Henry's law table or the Winkler test for dissolved oxygen. During the last 2 decades, a new form of electrode was developed based on the luminescence emission of a photoactive chemical compound and the quenching of that emission by oxygen. This quenching photophysics mechanism is described by the Sturmer-Eurovohmer equation for dissolved oxygen in a solution. The determination of oxygen concentration by luminescence quenching has a linear response over a broad range of oxygen concentrations and has excellent accuracy and reproducibility. There are several recognized EPA methods for the measurement of dissolved oxygen for B.O.D, including the following methods: standard methods for the examination of water and wastewater, Method 4500O, in situ incorporated. Method 1003-8-2009 Biochemical oxygen demand measurement by optical probe. Test limitations: the test method involves variables limiting reproducibility. Tests normally show observations varying plus or minus 10 to 20% around the mean. Toxicity: some wastes contain chemicals capable of suppressing microbiological growth or activity. Potential sources include industrial wastes, antibiotics and pharmaceutical or medical wastes, sanitizers and food processing or commercial cleaning facilities, chlorination disinfection used following conventional sewage treatment, and odor control formulations used in sanitary waste holding tanks in passenger vehicles or portable toilets. Suppression of the microbial community oxidizing the waste will lower the test result. Appropriate microbial population: the test relies upon a microbial ecosystem with enzymes capable of oxidizing the available organic material. Some wastewaters, such as those from biological secondary sewage treatment, will already contain a large population of microorganisms acclimated to the water being tested. An appreciable portion of the waste may be utilized during the holding period prior to commencement of the test procedure. On the other hand, organic wastes from industrial sources may require specialized enzymes. Microbial populations from standard seed sources may take some time to produce those enzymes. A specialized seed culture may be appropriate to reflect conditions of an evolved ecosystem in the receiving waters. History of the use of B.O.D, the Royal Commission on River Pollution, which was established in 1865, and the formation of the Royal Commission on Sewage Disposal in 1898 led to the selection in 1908 of B.O.D-5 as the definitive test for organic pollution of rivers. 5 days was chosen as an appropriate test period because this is supposedly the longest time that river water takes to travel from source to estuary in the UK. In its 6th report, the Royal Commission recommended that the standard set should be 15 parts by weight per million of water. However, in the 9th report the Commission had revised the recommended standard. An effluent taking up 2-0 parts dissolved oxygen per 100,000 would be found by a simple calculation to require dilution with at least 8 volumes of river water taking up 0.2 part if the resulting mixture was not to take up more than 0.4 part. Our experience indicated that in a large majority of cases the volume of river water would exceed 8 times the volume of effluent, and that the figure of 2-0 parts dissolved oxygen per 100,000, which had been shown to be practicable, would be a safe figure to adopt for the purposes of a general standard, taken in conjunction with the condition that the effluent should not contain more than 3-0 parts per 100,000 of suspended solids. This was the cornerstone 2030 plus full nitrification standard which was used as a yardstick in the UK. Up to the 1970s for sewage works effluent quality. The United States includes B.O.D effluent limitations in its secondary treatment regulations. Secondary sewage treatment is generally expected to remove 85% of the B.O.D measured in sewage and produce effluent B.O.D concentrations with a 30-day average of less than 30 mg per liter and a 7-day average of less than 45 mg per liter. The regulations also describe treatment equivalent to secondary treatment as removing 65% of the B.O.D. and producing effluent B.O.D. concentrations with a 30-day average less than 45 mg per litre and a 7-day average less than 65 mg per litre. Typical B.O.D. values: most pristine rivers will have a 5-day carbonaceous B.O.D. below 1 mg per litre. Moderately polluted rivers may have a B.O.D. value in the range of 2 to 8 mg per litre. Municipal sewage that is efficiently treated by a 3-stage process would have a value of about 20 mg per litre or less. Untreated sewage varies, but averages around 600 mg per litre in Europe and as low as 200 mg per litre in the US, or where there is severe groundwater or surface water infiltration in flow. B.O.D. biosensor: an alternative to measure B.O.D. is the development of biosensors, which are devices for the detection of an analyte that combines a biological component with a physicochemical detector component. Biosensors can be used to indirectly measure B.O.D. via a fast-to-be-determined B.O.D. substitute and a corresponding calibration curve method. Consequently, biosensors are now commercially available, but they do have several limitations such as their high maintenance costs, limited run lengths due to the need for reactivation, and the inability to respond to changing quality characteristics as would normally occur in wastewater treatment streams. For example, diffusion processes of the biodegradable organic matter into the membrane and different responses by different microbial species which lead to problems with the reproducibility of result. Another important limitation is the uncertainty associated with the calibration function for translating the B.O.D. substitute into the real B.O.D. B.O.D. software sensor: Rustam al. proposed the use of the KSOM to develop intelligent models for making rapid inferences about B.O.D. using other easy-to-measure water quality parameters, which, unlike B.O.D., can be obtained directly and reliably using online hardware sensors. This will make the use of B.O.D. for online process monitoring and control a more plausible proposition. In comparison to other data-driven modeling paradigms such as multi-layer perceptrons, artificial neural networks and classical multivariate regression analysis, the KSOM is not negatively affected by missing data. Moreover, time sequencing of data is not a problem when compared to classical time series analysis. See also, chemical oxygen demand, theoretical oxygen demand, carbonaceous biochemical oxygen demand. Wastewater quality indicators discusses both B.O.D. and C.O.D. as indicators of wastewater quality. Further references: Rustam R., A.J. Adaloy, and M. Skoltz applying Kohanen self-organizing map as a software sensor to predict the biochemical oxygen demand, Water Environment Research, 80, 32 €40. References. External links: B.O.D. Doctor, a troubleshooting wiki for this problematic test.